In this video, we will discuss about the Sialoglycans and the Siglec specificity variable. So basically, how the Sialoglycans specificity to a specific Siglec is determined, or what are the different variables on which the Sialoglycan specificity to a specific Siglec is depend on. So we will first start with this type of sialic acids. As whoever in the glycobiology field must be aware about this type of sialic acids, and there are three major kind of sialic acids, that is nu5AC, nu5GC and KDN. Out of this nu5ACs are the one that are presently forming the human beings. So based on the sialic acid, it will affect the Siglec specificity or sialoglycan specificity to this sialic acids. And the next things is this glycoconjugate composition. So basically, whether a glycans is composed of different kind of glycan monosaccharides, such as whether it's a high mannose sialic acid dependent glycan, or whether it's a hybrid sialic acid dependent glycans. This will also determine its specificity. This will also determines which sialoglycans is highly specific to the Siglec. It should also determine which Siglec is specific to this sialoglycans, or based on its compositions, what are its specific Siglec to which this composition of the glycans can bind. Other things is about the types of linkage. So basically, sialic acids can attach to the penultimate glycan through different kinds of linkage. It is alpha 2,3 linkage, alpha 2,6 linkage, or alpha 2,8 linkage. So these types of linkage will also affect the specificity of the sialoglycans and Siglec binding. And the next thing is about the modifications of the sialic acid. And not just sialic acid, but also the glycans that are involved in this, or the monosaccharides that are involved in this glycan. Here the acetylations of the sialic acid is shown, and also the sulfations of the this mannose and glycanac is shown. This will also overall modifications will also affect that which Siglec it is more prominent to bind. And afterwards, the backbone of the glycans, that is glycan is carried on which kind of backbone, whether it's a protein or this glycoprotein, or whether it's a lipid on the cell surface, or whether it has recently been found out that glycans are also found on the RNAs, whether it's RNA. So this backbone structures will also plays a role in this Siglec acid and sialoglycan interactions. Next thing is about the types of glycan, whether it's O-linked glycan or it's an N-linked glycan, and that has also shown in the previous experiments, or in the researchers has also found out that based on their linked with the backbone protein, whether it's an N-linked glycan or O-linked glycan, it will also has an impact on this Siglec acid and sialoglycans binding. And afterwards, it's about this distributions of the glycosylation site. If the glycosylation site has two clustered, then it might affect one glycan site might affect the interactions with the other glycan site. So the distribution of glycosylation site also plays a role. If they are too close, it might cause a hindrance effect or some other kind of resistance. And apart from that, the complex that it forms with the other proteins, the glycans are also known to form interactions with the other protein. If they form interactions with the other protein, then it might also plays a role towards the sialoglycans and Siglec specificities. And some kind of anchors favors some kind of sialoglycans and Siglec acid bindings, and some doesn't favor. This anchor from the protein context will also determines to which sialoglycans it favored, and to which Siglec it favors and which Siglec it doesn't favor. So these are some of the variables the common variable. These are some of the prominent variables that are found to affect the Sialoglycans and Siglec specificity. So it is not just a one-on-one interactions. It will also depend on the surrounding factors and also the different compositions, even the linkage, how the sialic acid linked to the other glycans. So this is just a brief overview of what are the major variables on which sialoglycans and sialic acid specificities depends. I hope you understood these variables to some extent. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.